Very good evening students. Here we are to discuss all the NEET previous year questions from two major chapters. Those are photosynthesis in higher plants and respiration in plants. Right? We are going to discuss all the questions from the year 2024 to the year 2019. Yes, if we look into the most recent question which is asked on the energetics, okay, the most recent question is asked on the energetics of Kelvin cycle, wherein how many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in Kelvin cycle. If you look Kelvin cycle, Kelvin cycle, where we all know Kelvin cycle is operated in 3 steps, right beta? What are they? Carboxylation, reduction and regeneration. And what ma? Regeneration. Okay. For every molecule of carbon dioxide that is fixed, 2 ATP and 2 NADPH are utilized in reduction step, 2 ATP and 2 NADPH are utilized during reduction, and 1 ATP, 1 molecule of ATP are utilized in regeneration for every carbon dioxide fixation for each cycle of Kelvin cycle. On total, how many ATP ma? On total 3 ATP and 2 NADPH. It is 3 molecules of ATP and 2 molecules of NADPH. Option 1 is the correct answer. And second question, if you see, that is a statement based question where it is again on the light independent reactions. In statement 1, he is saying like in C3 cycle or in C3 plants, some oxygen binds with rubisco, hence carbon dioxide fixation is decreased. Yes, during high light intensity or high temperature conditions, what happens? Rubisco in C3 plants has equal potential to bind both with carbon dioxide and oxygen, right? Ribulose, bisphosphate, carboxylase oxygenase enzyme has equal potential to bind with carbon dioxide and oxygen. So whenever oxygen quantity is more and carbon dioxide concentration is less, then oxygen binds with rubisco, which activates the process of photorespiration. This activates the process of what beta? Photorespiration, thereby carbon dioxide fixation is decreased. Yes, of course, statement 1 is true. If you look into statement 2, in C4 plants, mesophyll cells show very little photorespiration. Wrong. In C4 plants, rubisco enzyme is not present in the mesophyll cells. That is why photorespiration does not conduct only in the C4 plants. Okay. So, statement 2 is wrong. If you clearly look into the options, statement 1 is true and 2 is false. Option 4 is the correct answer. Clear ma? Yes. We are going to exercise the concept and the objectives. Very important session. Stay focused beta. Yeah. If you look into the next question, which of the following are required for dark reactions of photosynthesis? Dark reaction of photosynthesis will require, dark reaction means light independent reactions, right, which takes place where beta? It takes place in the stroma of chloroplast, where carbon dioxide is fixed or reduced to glucose, right, where C3 cycle is operated in C3 plants, C3 and C4 is operated in C4 plants, C3 and CAM cycle is activated in CAM plants. For this, what is required? Of course, carbon dioxide is required, ATP and NADPH, which is the assimilatory power, which is what ma? Assimilatory power, which is formed at the end of light reactions. Both ATP and NADPH, which are formed as the products of light reactions, are utilized by dark reactions. So, we need carbon dioxide, ATP and NADPH. So, chlorophyll and light are not required. These are required for light reactions, not dark reactions. So, what will be our answer here? C, D and E. Option 4 is the correct answer. Next, Which of the following are required for light reaction of photosynthesis is asked. Okay, this is asked in the NEET 2024. Okay, so yes, for light reaction we need oxygen, water for photolysis, chlorophyll and sunlight. All from B, C, D, E, all these are required for light reaction of photosynthesis. Sorry, oxygen is not required. Oxygen is not required. We require chlorophyll, light and water. All these three are required. Oxygen is required for cellular respiration or aerobic respiration in mitochondria. This is not required for light reaction. So yes, the option is C, D, E. Option 2 is the correct answer. Next question, if you see, which one of the following products diffuses out of the chloroplast during photosynthesis? During photosynthesis, NADPH and ATP, they cannot diffuse out of the chloroplast because they are present in the stroma and we need them. Okay, ADP is also present in stroma for the process of photophosphorylation. Okay, oxygen will diffuse out of the chloroplast. It is what oxygen that diffuses out of the chloroplast. Fine. So option 3 is the correct answer. Next, sixth question, which is also appeared in NEET 2024, which says, like, the question is asking about, observe the figure, identify different stages labeled in the alphabets by selecting. This is the image based question. The question is over here. Here if you see, based on this image, he is asking us to identify what is A, B and C. It is based on what Kelvin cycle. See how important the Kelvin cycle is. Okay, if you see it clearly, first step of Kelvin cycle, which is the most crucial step, that is carboxylation. The first step is what, Nana? Carboxylation is the first step. Next is what? Next, second step is reduction, where 2 molecules of 3-phosphoglyceric acid is reduced to 2 molecules of glyceraldehyde 3-phosphate. And the third step is regeneration. Third step is what, regeneration. So here A is carboxylation, B is reduction and C is regeneration. Option 3 is the correct answer. Okay. Next, if we look into, how many ATP and NADPH2 are required for the synthesis of 1 molecule of glucose in Kelvin cycle? We all know for the formation of 1 molecule of glucose, we need 6 carbon dioxides which conduct 6 Kelvin cycles. How many Kelvin cycles, 6 Kelvin cycles. We all know for 1 carbon dioxide fixation, just now we have learned, we need 3 ATP and 2 NADPH, right? 3 ATP and 2 NADPH. For 6 molecules of carbon dioxide, we require 6 into 3, that is 18 ATP. 6 into 3, how much, 18 ATP and 12 NADPH, and we require 12 NADPH. Clear? So here what is the answer? 18 ATP and 12 NADPH. Next, coming to the question which is asked in NEET 2023, the reaction center PS2 has an absorption maximum of what? We all know PS2 absorption maxima is 680 nanometers and PS1 absorption maxima is 700 nanometers. Okay, photosystem 1 is 700 nanometers, 2 is 680 nanometers. So what will be our answer, beta? Option 4 is the correct answer. Next, given below are 2 statements. Again a statement based question in 2022. Okay, so here in statement 1, if you see, the primary carbon dioxide acceptor in C4 plants is phosphoenol pyruvate. Okay, PEP is the primary carbon dioxide acceptor in C4 plants and found in the mesophyll cells. True. Mesophyll cells of C4 plants lack Rubisco enzyme. Correct. Both the statements are true. Statement 1 and statement 2 are correct. Option 4 is the right answer. Next question, if you see, which one of the following is not true regarding the release of energy during ATP synthesis during chemiosmosis? Chemiosmosis process requires what is the question, which is not true. Not true regarding chemiosmotic hypothesis he is asking. First is breakdown of electron gradient, second movement of proton across the membrane, third reduction of NADP to NADPH2 in the stroma side of the membrane, breakdown of proton gradient. Which of the following is not true regarding the release of energy? Okay, we have three major reasons, right? Three major reasons. One is during Z scheme, PQ is accepting protons from stroma and giving it to the lumen. Through PQH2, the H plus are transferred to thylakoid lumen. Second is photolysis of water. Second is what, ma? Photolysis of water. And third is NADP plus is reduced to NADPH plus H plus by the enzyme NADP reductase. All these three steps are the crucial steps which is leading to the proton gradient. Okay, more proton accumulation in the thylakoid lumen and less H plus in the stroma. Less H plus in the stroma, which is activating the proton gradient and leading to chemiosmotic hypothesis. This all of us know. Yeah, this is the breakdown of proton gradient, correct? Reduction of NADP plus to NADPH2, correct? Movement of protons across the membrane, correct? Breakdown of electron gradient, wrong. It is the breakdown of proton gradient, not electron gradient. Okay. So yes, this is option 1. Clear, ma? Next. Yeah. What is the role of large bundle sheath cells that are found around the vascular bundles in C4 plants? In C4 plants, you see, whenever we talk about C4 plants, immediately that comes to your mind is what, Nana? Kranz anatomy, right? What that comes to your mind? Kranz anatomy, wherein vascular bundles which are arranged in the mesophyll cells. Around this vascular bundles, you can find bundle sheath cells which are arranged in the form of wreath. Okay, bundle sheath cells which are arranged in the form of wreath without any intercellular spaces. And this bundle sheath cells, Kranz anatomy. Yeah, and this bundle sheath cells have agranule chloroplast. What does they have, beta? Agranule chloroplast. Okay, grana is absent in them. And inside stroma, what enzyme is present? Rubisco is present, which will conduct C3 cycle. This will conduct C3 cycle. Clear? So here this bundle sheath cells are for what purpose is the question. Is this to increase the number of chloroplast for operation of Kelvin cycle? Correct. To enable plant to tolerate high temperature? Wrong. To protect vascular tissues? Wrong. To provide site for photorespiratory activity? No, this is an adaptation to prevent the photorespiratory activity. Okay. So, bundle sheath cells which are present around the vascular bundles, their main activity is to conduct C3 cycle or Kelvin cycle. Clear? And they have what type of chloroplast, beta? Agranule chloroplast. Keep that in your mind. Next, coming to 2021 question, which of the following statements is incorrect? He is asking. Incorrect statement. Stroma lamellae has PS1 only and lack NADP reductase. Yes. Stroma lamellae has only PS1 and they lack NADP reductase, correct? Grana lamellae has both PS1 and PS2, true. Cyclic photophosphorylation involves both PS1 and PS2, wrong. Both ATP and NADPH plus H plus are synthesized during non-cyclic photophosphorylation, correct. So, here whenever statement based question is asked, look into the question properly whether they are asking you the incorrect statement or correct statement. So, here if you see statement 3 beta, cyclic photophosphorylation is involves two photosystems, that is PS1 and PS2. Two photosystems are involved in cyclic photophosphorylation, which is seen in bacterial photosynthesis. Bacterial photosynthesis, wherein in stroma lamellae only one photosystem is present, and that conducts cyclic photophosphorylation. Two PS are not present in the cyclic photophosphorylation, only one photosystem is present in the stroma lamellae of bacterial photosynthesis. Clear? Is that fine? So, here incorrect statement is what ma? 3. Next, if you see, question that is asked in 2021, wherein the first stable product formed in carbon dioxide fixation of sorghum. You have to remember these examples ma, C4 plants, okay, C4 plants, best examples you can remember like SAM, interesting, right? Sorghum, maize, sugarcane, sorry, it is SMS. SMS, shortcut to remember C4 plants, that is sorghum, maize, sugarcane. Sorghum, maize and sugarcane. Simple trick to remember C4 plants, like you know, yeah, sorghum conducts what type of fixation? C4 cycle in the mesophyll cell and C3 cycle in the bundle sheath cells. What is the first stable product formed in the C4 cycle beta? Oxaloacetic acid, which is a 4-carbon compound. Clear? Next, which of the following is not a product of light reaction of photosynthesis? ATP and NADPH are formed. Oxygen is formed due to the photolysis of water. So, all these three are the products of light reaction except NADH. So, option 2 is the wrong, option 2 is the answer here, because NADH is not the product of light reaction. NADPH and ATP are the products, okay. They are the assimilatory power formed at the end of the light reaction, and oxygen is formed as a result of photolysis of water in the light reaction. Clear? These are the PYQs from the chapter photosynthesis in higher plants. Next, if we look into we will also exercise the PYQs of respiration in plants, okay. In respiration of plants, if you look, most of the questions asked are match the following type of questions. Most of them are what type beta? Match the following type of questions. You have to analyze them carefully. First is, yeah, citric acid cycle. Sorry, this has exchanged. In list 1, option 1 is what? Citric acid cycle, which is nothing but TCA cycle, okay. In 2, they have provided you the locations. 2, they have provided you the locations. Where does that takes place? Citric acid cycle takes place, or TCA cycle will take place in mitochondrial matrix. Mitochondrial matrix. Okay, citric acid cycle will take place in mitochondrial matrix, that is 2. Okay, TCA cycle takes place in mitochondrial matrix. So, A is 2, and then glycolysis. Glycolysis takes place in the cytoplasm. B is 1. Glycolysis takes place in cytoplasm, and then electron transport system ETS takes place in inner mitochondrial membrane. Okay, ETS takes place where beta? Inner mitochondrial membrane. C is 4. D, proton gradient. Proton gradient is generated in intermembrane space. Proton gradient is generated where beta? In the intermembrane space. For your proper understanding, once let me give you a quick view on the aerobic respiration. Okay, glycolysis takes place in the cytoplasm. At the end of glycolysis, 2 molecules of pyruvic acid are formed. Due to the incomplete oxidation or partial oxidation of glucose, 2 molecules of pyruvic acid is formed. This 2 molecule of pyruvic acid will directly go to the mitochondrial matrix. It will directly go to where? Mitochondrial matrix. Inside the mitochondrial matrix, TCA cycle or citric acid cycle is operated. Okay, oxidative decarboxylation and TCA cycle are done in the mitochondrial matrix. Later, ETS is done in the intermembrane space. Because of the ETS, proton gradient is generated in between intermembrane space and mitochondrial matrix. Proton gradient is generated in the intermembrane space. Clear beta? How you have understood glycolysis in the cytoplasm, TCA cycle and oxidative decarboxylation in mitochondrial matrix, electron transport system in the inner mitochondrial membrane, proton gradient is generated in the intermembrane space. Clear? Yes. Next, if we look into again a question in the year 2024, wherein identify the step in TCA cycle which does not involve oxidation of the substrate. Which doesn't involve oxidation of substrate. Isocitrate to alpha ketoglutaric acid, decarboxylation reaction, oxidation happens. Malic acid to OA, which is the last step of TCA cycle, oxidation is involved. Succinic acid to malic acid also oxidation is involved. If you look succinyl CoA to succinic acid, which is substrate level phosphorylation, which releases GTP or ATP. Okay, this is not an oxidation. Here there is no involvement of oxidation of substrate. So option 4 is the correct answer. Again, if you can observe, match the following question which is appeared in the NEET 2024, wherein NADP dehydrogenase is complex 1. ETS, just now we have discussed, ETS operates in inner mitochondrial membrane, proton gradient is generated in intermembrane space. So here NADH dehydrogenase is complex 1. Then complex 2 is succinate dehydrogenase. Complex 3 is cytochrome B, complex 3 is cytochrome BC complex. Complex 4 is cytochrome C oxidase. So here A is 1, B is 2, C is 4 and D is 3. Option 4 is the correct answer. Next, if you consider, again match the following type of question. So if you see respiration in plants, many a times the question pattern is match the following type of questions only beta. So exercise most of the match the following questions from this chapter. Okay, that is my advice. Again, if you see oxidative decarboxylation, in list A he has given us some processes, in list B he has given us the enzymes. Okay, oxidative decarboxylation includes the enzyme pyruvate dehydrogenase. Oxidative decarboxylation is also called as what ma? Link reaction or transition reaction. Link reaction or transition reaction. Okay, link reaction or transition reaction, wherein pyruvic acid which is just landed into the mitochondrial matrix, pyruvic acid which is just landed into the mitochondrial matrix is converted to acetyl CoA. Is converted to what beta? Acetyl CoA by oxidative decarboxylation. Okay, where pyruvic acid is combined with a coenzyme A and NADP plus, which results in the formation of acetyl CoA and NADH plus H plus. Clear? So yes, this enzyme, the enzyme which is required over here is pyruvate dehydrogenase. Glycolysis is also called as EMP pathway, which is named after the scientists Emden, Meyerhoff and Parnas. Then oxidative phosphorylation where is nothing but ETS, okay, electron transport system, which is done in inner mitochondrial membrane. TCA cycle occurs in A TCA cycle involves the enzyme that is citrate synthase. The very first enzyme is citrate synthase. So, if you clearly see the options, A is 2, B is 3. Okay, here the options are not given, I will let you know. A is 2, B is 4, yeah, here it is given, C is 3 and D is 1. So, option 3 is the correct answer. Here the correct answer is what beta? Option 3. And next, yeah, here it is assertion reason question, assertion reason question. So, mostly are getting match the following, statement based and assertion reason questions. Exercise more such type of questions from this chapters ma. So, assertion reason, if you see, in assertion ATP is used at 2 steps in glycolysis. True. At what steps ma? At step 1, first step is used in converting glucose to G6P, glucose 6 phosphate, by the enzyme hexokinase. By what enzyme Nana? Hexokinase. And second ATP is used for the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. Okay? From fructose 6 phosphate to fructose 1,6 bisphosphate, that is third step of glycolysis, which is mediated by the enzyme phosphofructokinase, where this step is called as rate limiting step. This step is called as what Nana? Rate limiting step. Clear? So, here we can consider the option that is A is correct and R is true. R is explaining assertion. Okay, that will be the correct answer. I did not put the options, but the correct answer is assertion and reason are true, and here reason is explaining the assertion. Clear? And 2023, it is about chemiosmosis again. If you can see from chemiosmosis, the questions are asked. What is the correct combination of chemiosmosis? Chemiosmosis requires membrane, proton pump, proton gradient and ATP synthase, which has F0 and F1 pump. Okay? So, option 4 is the correct answer. Chemiosmosis requires membrane, proton gradient, proton pump and ATP synthase. And then, what is the net ATP when each molecule of glucose is converted to 2 molecules of pyruvic acid? So, here if you look carefully beta, in glycolysis process, look carefully, glucose undergoes 10 biochemical reactions, okay, 10 successive steps takes place. At first step, you know, 1 molecule of ATP is utilized. At third step, again 1 molecule of ATP is utilized, okay. At sixth step, 2 molecules of NADH are released. Again at 7th step, 2 molecules of ATP are released. At 10th step, 2 molecules of ATP are released. These are the energetics, right? Out of 4 ATP that are released into the cytoplasm, 2 ATPs are required for the steps, okay. First and third step are utilizing 2 ATP out of 4 ATP that are released in the payoff phase, right. So, yes, here the net ATP of glycolysis will be, 2 ATP. It is 2 ATP because from the 4 ATP released, 2 are utilized by the preparatory phase of glycolysis, okay. That is why the net ATP is 2 ATP. If the question is what is the total energy of glycolysis, what is the total energy of glycolysis, if that is the question, then our answer should be 2 NADH. We know approximately 1 NADH is equal to 3 ATP. So, 2 into 3, 6 ATP plus 2 ATP, 6 plus 2, it is 8 ATP. Okay, total energy of glycolysis is 8 ATP, net ATP of glycolysis is 2 ATP, that you have to remember beta. So, here what is the net gain of ATP for each molecule of glucose is 2 molecules of ATP. Done? Clear with this? Next, what is the net gain of ATP when each molecule of glucose is converted to the same question? It is 2 molecules of ATP. Then if you look into statement based question again asked in the year 2021, which of the following statements is incorrect, he is saying. Okay. In electron transport chain, one molecule of NADH gives rise to 2 ATP. Wrong. One NADH, just now we have discussed, gives rise to 3 ATP, not 2 ATP. And one FADH2 gives rise to 2 ATP, not 3. So, he is asking incorrect statement, then our answer will be option 1. Second option if you see, ATP is synthesized through complex 5, correct. Oxidation reduction reactions produce proton gradient in respiration. Proton gradient between what ma? Intermembrane space and mitochondrial matrix. Then during aerobic respiration, role of oxygen is limited to the terminal stage. Yes, in ETS, what is the terminal acceptor of the electron or what is the ultimate acceptor of the electron? That is oxygen. Oxygen is the ultimate acceptor of the electron. Yes, understood? Yes. Now, if we look into the last set of questions from this chapter, the oxygenation activity of Rubisco enzyme, in photorespiration leads to the formation of, okay, oxygenation of Rubisco in, that is C2 cycle which takes place only in the C3 plants. What is the formation of the oxygenation of Rubisco enzyme in photorespiration leads to what formation he is asking. Look here carefully. Oxygen molecule, when it binds with RUBP, which is a 5 carbon compound, in the presence of the enzyme Rubisco, what happens is one molecule of 3-GP, okay, glyceraldehyde 3-phosphate, phosphoglyceric acid, one molecule of phosphoglyceric acid and one molecule of phosphoglycolate, which is 2 carbon, this is 3 carbon is formed, okay. One molecule of phosphoglyceric acid and one molecule of phosphoglycolate are formed. So, here because of the oxygenation, one molecule of 3 carbon compound, that is phosphoglyceric acid, is formed, correct. One molecule of 6 carbon, wrong. One molecule of 4, wrong. 2 molecules of 3 carbon, wrong. So, option 1 is true. It forms one molecule of phosphoglyceric acid and one molecule of phosphoglycolate. And then next question, if you see, conversion of glucose to glucose 6-phosphate is the first irreversible reaction of glycolysis. Okay, glucose to glucose 6-phosphate is the first irreversible reaction. It is catalyzed by the enzyme hexokinase. Catalyzed by what enzyme beta? Hexokinase. And last question which is asked in 2019, if you see, it is based on the respiratory quotient. It is based on what, Nana? Respiratory quotient. Respiratory quotient is equal to amount of carbon dioxide consumed by oxygen evolved. Okay, that is the respiratory quotient. For tripalmitin means fats. RQ value for fats will be less than 1, that is 0.7. Let us recall the RQ value of other respiratory substrates too. For carbohydrates, RQ value will be 1. Okay, 6 carbon dioxides by 6 oxygen, it will be 1. For fats, as we have discussed, it is 0.7. For proteins, it will be 0.9. Okay, and for fats and for proteins and for organic acids, it is greater than 1. Organic acids, it is greater than 1. For anaerobic respiration, for let us consider for alcoholic fermentation, where in alcoholic fermentation 2 carbon dioxides will be released, right? 2 carbon dioxides by no oxygen, 2 carbon dioxides by no oxygen, 0 oxygen, something by 0 is infinity. Okay. So, in an alcoholic fermentation, RQ value is infinity. These are few RQ values you have to remember for sure. Clear, ma? So, this and all are the questions and the concepts that are revised based on the PYQs of 2 chapters, that is photosynthesis and respiration. In coming up sessions from tomorrow, we are going to start the sessions on all the previous year papers on 2019 botany paper, 2020 botany, zoology, physics, chemistry. All the question papers with the concepts will be dealt in a more detailed way. So, do not miss those sessions which are highly useful for you people. Clear? Thank you all. Meet you tomorrow.